Please go ahead, Dr.
Okay.
Sahu. It's
Yes,
visible.
sir. It is
Okay.
visible.
Okay. So, I changed the slide. Can you see that the slide is changed?
Yes, yes, we can
Yeah.
see.
Yeah. Okay. So, basically, I'm going to summarize about COVID-19. Um, so the coronavirus disease, 2019, and what we call shortly as a COVID-19, we all know that is caused by a novel coronavirus, which we now named as SARS-CoV-2 because there was already SARS-CoV-1 earlier, which disappeared mysteriously. So what the coronavirus, the first question is why the name coronavirus? And in Latin, the word corona means actually the crown. And since these viruses have spikes which look like spikes in the crown, they are called coronavirus, and that's how the structurally they are defined. You can see the structure which is shown here, the EM structure where you can clearly see the spikes. So coronaviruses basically are zoonotic viruses. What it means basically that these are there in the animals and they jump the species and they start infecting humans. So this has been happened earlier. And the peculiar thing about coronavirus, that it causes respiratory distress, and this particular one causes really, a lot of problem all over the globe. The symptoms what we get, as we all know now, very well publicized in the media, that the symptoms are pretty much like flu. So we get fever, headache, muscle ache, cough, sore throat, and breathlessness. So these are the common symptoms. There are many more other symptoms which you see in severe patients, but these are the normal common symptoms which we see. So where did it all start? So this started in Wuhan in something called the Hunan fish market or Hunan wholesale fish market. I think this is a misnomer, the name fish market, because I think it seems that pretty much everything that moves is sold in this place. And you can see that there very little section, one side is a fish, but you have everything else which is also sold, including bats and everything, you can name it. So the whole story really started here. So let's look at the timeline and the origin and the spread of the virus. So the first case of this mysterious coronavirus or SARS-CoV-2 actually happened in December 8th. And on December 31st, the China alerted the WHO about this mysterious pneumonia-like disease. And that's when the whole world came to know that something is going on in Wuhan and there is some mysterious disease. Later on, this was not just limited to Wuhan, it moved out of the China, and the case was reported from in the Thailand and on Jan 13th. Now, after this was reported, and getting again and again, WHO immediately declared this as a global public health emergency on January 30th, and that's when we also in India got the first case in Kerala. Now they started actually moving into different countries, and more cases started, in countries started reporting. And this was almost reported in 100 countries with over 100,000 cases, and on March 11th, then WHO declared this as a and that now this is global, this is everywhere, and this is becoming pandemic, and they declared it as a pandemic. So as of 20th July, we have over 14 million cases in 188 different countries, over 500,000 deaths. But the good thing is that a lot of people are recovering. So there are recoveries almost more than close to 8 million people, which is a good sign. So let's look at the timeline in India. So as I said, it got first introduced on Jan 30th in Kerala. On March 2nd, we also had cases in Delhi and Hyderabad. And then slowly, this started more cases, and we then, our Prime Minister declared Janta curfew on March 22nd and a complete shutdown for 21 days, which was declared by the Honorable Prime Minister on March 24th, which went on till the end of May actually. And we then started opening up slowly. And currently we have over 1 million cases, and out of which 4 lakhs are still active. We have good recovery, about 7 lakh people recovered, but unfortunately more than about 27,000 people died during this outbreak. So as of 20th, if you see, we have many confirmed cases, and a lot of cases now. Maharashtra has become a hub. So yes, we have a lot in Maharashtra, also in Delhi and Tamil Nadu, and now, uh, of course, Bangalore and in Karnataka is also increasing. So there are many active cases, close to 4 lakhs now, but good thing is that the recovery rate is pretty good, and about 63%, and over 7 lakh people have recovered. So the question obviously is, why this disease really became pandemic. I mean, coronaviruses are there, they are zoonotic viruses, they do jump, and they have been jumping, and we know earlier MERS and SARS-CoV-1, and also there are seasonal coronaviruses which you see in Hong Kong. So why this, what was so peculiar about this particular virus that it became pandemic? So I tried to jot down few things. So the first and primary reason obviously is this is a novel virus. We were not exposed to this particular virus, and our body did not have any pre-existing defense against this particular virus. And therefore, once we get exposed, there is nothing which we have pre-existing immunity, and therefore it starts flaring up. The second important reason is it's airborne. So people who are carrying, and when they are traveling from one place to another place, and particularly now world has become a global village. So people are traveling across the globe and they're carrying this virus, so it's very easy to spread. And the respiratory viral diseases are one thing which spread. And last time, we came across H1N1, which got, everywhere. And now we have the coronavirus. So respiratory viruses are very easy to spread, and this belongs to that particular category. The third thing is it has very high transmission rate. It transmits very easily anywhere. One person can give it to around 3 people is what we, has been estimated. So the transmission rate is again very good, which also makes it that it can spread very easily. The fourth point is long incubation period. People, when they are infected, it can be anywhere from 1 to 12.5 days. And so they are incubating, they are traveling, and they don't know that they have this infection. And therefore, they are carrying to different places. And that's how the virus can move from one to another place. The another problem, obviously, is asymptomatic transmission. We know that many people have asymptomatic cases, they have the virus. Again, in this particular cases, we have seen during, when we are testing that the load is pretty high. It is not that asymptomatic people carry less load of the virus. The load is pretty high actually in some, in many cases, much more than even people who are actually symptomatic. So they are carrying and they are sort of, giving the virus to other people. So that's another. So together, this has made it a pandemic. So these are the main points which I have come across. And if you compare the SARS-CoV-1 versus SARS-CoV-2, you can see that the SARS-CoV-1 was not, it was more pathogenic. This one is less compared to SARS-CoV-1, but you can see the number of cases, there is a steep rise, and this went everywhere very fast. So let's look at the numbers, particularly for the SARS-CoV-2. We all know that when we talk, when we cough, when we sneeze, the virus is liberated in pretty high concentration, right? So even when we're talking, we are actually giving away the virus. But is there any other route where the viral load is very high and the stool, uh, the load is again very high? So, what does it really mean that when we talk, uh, and how many particles are really needed to get infected? And as low as 1,000 SARS-CoV, people believe, and it has been estimated that scientists, they are enough to make, infect. And what it means to translate that if you are talking to a person who is, infected for 10 minutes, then you can get this infection. Okay, face to face, if you're talking to a person for 10 minutes, you can get this infection. Scientists also looked at the environmental stability of this virus, that now we know, it is can go through aerosol also, which means that when the size is less than about 5 micron, then they are not called as droplets, more like aerosols, smaller ones, and they can hang on in air for almost 3 hours, right? So if it is a closed rooms, then they can hang on. If it is an open area, obviously it will get diluted. So the roughly half-life in the aerosol of this virus is about 3 hours, which was published in New England Journal of Medicine. Uh, what about the surface? We always say that surface are contaminated, one should not touch it. How long they really stay? So if you're talking about plastic or the stainless steel surface, then they can hang on there for almost 72 hours. They are infectious for that long. Copper somehow, they are not so good. So they don't hang on so long in copper, only 4 hours. But cardboards and stuff like that, they can stay almost a day, 24 hours. Okay, so they are there for quite a while when it comes to the surface, almost up to 72 hours. So that is something, the number one needs to keep in mind. So what about the virus? How does it look? As I said, the name coronavirus itself means that it has, spikes, crown-like spikes. So these spikes are basically trimers. They are trimers of spike protein. And the peculiar thing about this, that the affinity in this particular virus, the SARS-CoV-2, is very high. It's in nanomolar, anywhere from about 40 nanomolar, which is much higher than the earlier SARS-CoV-1. So this is, one of the reasons why, it infects so well. Another thing is, the question is how big the size of the particle is. It's around 100 to about 120 nanometer in size. So that's something like 1/100th of a hair, thickness of the hair. So that's the size roughly of the cell, which is pretty big, but it is almost same as what one sees like a flu virus. The size is pretty much same. If one compares, the genome of this and looks into what about the other coronaviruses like SARS-CoV-1 or MERS or the common cold, then what you see is that it is more closer to the bat CoV, but it's still not, same. It's only 96%. What it means that what is predicted is that when it jumped from bat to human, there was an intermediate species. So it went into some intermediate species, and from there it jumped into human. What that intermediate species, we still don't know, and people are still looking into that. So is virus changing? And that's something one would like to know. So this is a RNA virus, right? And RNA viruses are known to mutate 1,000,000 times higher than the host. So they mutate a lot. So what about the coronavirus? Is it mutating? It is mutating. It has a proofreading activity, so the mutation rates are a little lower, but it is mutating and the rates are lower. It's about 4 times lower than the flu virus, which we know very well. So the mutations are there, but the rate is not that high. But it is mutating, and almost in different places it is mutating. So one of the theory which people always think and discuss and talk is that when the virus comes and stays for a long time, it becomes less and less pathogenic. It gets mutated and becomes less pathogenic. So far, we have not seen that the virus SARS-CoV-2 is getting less pathogenic. The mutations, which are particularly in RDRP and S protein, and these are linked mutation. There are 4 nucleotide variations which the world has seen, and these are linked. And 2 are the ones which change amino acid. One is in RDRP and another is in the S protein. And this protein change is at the 614, which is quite talked nowadays about. If anything, this has made it little more pathogenic. The transmission people have seen is more, and the infection rate is more when you get, this mutation from ASP to GLI. And what we see overall, that the pathogenicity so far doesn't seem that it is going down. So how do we test that a particular person is exposed or infected with the virus? The most common is directly looking at the virus, and what we look directly primarily is looking at the viral DNA by the RT-PCR test, and pretty much everyone now knows about the RT-PCR test, where we use different probes and we try to see if these genes are there, and if we see, more than 2 genes. So we initially look for each gene. We see more genes. We say that it is positive. The other test, which is more to do with the point of care test, is the antigen test, where we directly look for the antigen. Now, this one is not as sensitive as the RT-PCR test, but if the load is higher, particularly in SARI cases, people see that always they will do antigen and it comes out positive, as what has been told to me by physicians who are directly dealing with these patients. So antigen positivity test is also another test, which is more like a point of care test. But if it is negative, then still one needs to go for doing RT-PCR to make sure that the person is negative. The third one is the antibody test, much talked about. This doesn't really tell that the virus is there or not there, but what it tells you is that the person has been exposed to this particular virus. There is a prior exposure. Whether it's active infection, it's not, you cannot say based on the antibody, but what you can say that and this is more for getting, you want to know what percent of population is getting infected is pretty good way of looking at it. And that is what India is also doing. So we all know that, most cases of these are asymptomatic, or they have very mild infection, which means that the people don't even know that they are actually suffering from COVID. And this is almost 80%, which is a very good scenario. The only problem is that since these people don't know, they can transmit it very easily to other people. The remaining 20% by and large require hospitalization, and they have to see the doctor, they have to be there, and then there are multiple. And 10% of this, they become really sick, and they need various support. And some percent, anywhere from 1 to 3%, die from this, and they become really, really sick. And in many cases, and people, we do understand a little bit about what is causing the severity, but there are still unknowns which we don't really understand. So the mild symptoms, everyone knows what the mild symptoms are, right? And when the symptoms become more shortness of breath, you have the oxygen level started dropping down, and one needs to worry about this. So fever, if you have a fever, high fever and shortness of breath, you must see a doctor. And it has been told again and again that one should look for the oxygen levels, and see if it is dropping more than 95, and it's critical when it's less than 90. But one, physicians say that if you're dropping less than 95, one should immediately be alarmed and see the doctor. So what is happening in severe cases? So once we get viral infection, our body tries to protect us, from the infection. So basically the innate immunity, which is much talked about, gets kicked in. And this is a non-specific immunity, which we try and, tries to protect us. And the specific immunity comes into play around, say, day 10 and after that. And, during this process, various cytokines and chemokines are produced, which are actually there to help our body to make this specific immune response. But what happens that sometimes they become very aggressive and they start destroying. So one of the reason in severe cases that something called cytokine storm, which is there, which is generated, and that is actually making it more severe cases. So this has been reported very well in the literature. So what are the treatments? Mild cases, pretty much everyone knows hydroxychloroquine is something which has been said that, and has been given. Favipiravir is also given, and these are the mild cases. So by and large, you treat. Uh, in moderate cases, the problems are becoming more. Remdesivir is suggested, hydroxychloroquine or the combination of both, but also the steroids. And we know that, there small clots are getting formed in patients, which has been reported. Initially, we didn't know about this in this disease, but later on people found. So anticoagulation and also the convalescent plasma, and much talked about now, is something which is given in moderate. In severe cases, I said like cytokine storm is there, right? So the monoclonal antibody against the IL-6, something called itolizumab, very difficult to pronounce. I don't know why people give these kind of names, which are difficult to pronounce. I don't think even the B cell people like this kind of names. But anyway, uh, itolizumab, if you don't remember, just is the antibody against the IL-6 receptor, which is there to block the cytokine storms. Other things which are given, say, steroids, anticoagulation, treatment, and so on and so forth. And these are some things which are there. Of course, it depends on the condition of the patients and what symptoms are, and the physicians take a call based on that. I'm not an expert in this, but I just wanted to introduce is how the treatment is done. So the bigger question is it here to stay? And the short answer is yes. So initially, people talked about that maybe there's a herd immunity, and we'll get the herd immunity, and therefore we'll be protected. And this, it turns out that one person can give it to 3 people, and if you do the math, about 66% of the population must be exposed to this, and that's a lot. And if we wait for that, a lot of fatalities will be there. So the question then is, how this is going to be contained, or will it be contained, or basically, what about the other coronavirus? What did we hear, right? And what do we learn from those? So, and are they still there? So for example, the MERS was another coronavirus, which is much, much more fatal than the SARS-CoV-2. And this is, there is still limited circulation for this. SARS-CoV-1 somehow disappeared mysteriously, and we don't understand how that disappeared. And there are also seasonal coronaviruses like flu, which happen every day, something like Hong Kong. And these are there. So what are the factors really, which determine the fate of any virus? One obviously is the environmental effect on the viral stability. Is it stable? How stable? This looks pretty stable. Transmission rate, this is very high again in this case. Host behavior, how do we behave? How do we try and contain this virus? What precautions we are taking will determine a lot on this. And changes in the immunity level over time, either by herd immunity or more importantly by vaccination. And that's what is going to decide, whether in how long this is going to stay. But my gut feeling is that this will become more like a seasonal infection, what is there about seasonal coronavirus, what we see in Hong Kong. probably more like that is what is going to happen. So will it be contained? India obviously sees a lot steep rise. If you see from the graph on your left, the something which is going up is India. But the good news is that it did get contained in other places. For example, Spain, Germany, Italy, uh, these places, if you see, there is a plateau. So there is a peak and followed by plateau, and we are still haven't reached the peak. So unless we reach the peak, and then it will start going down, but we haven't reached, which means that we are still, increasing in this. So what is going to help? More testing, more isolation, and more treatment. But it is going to get contained. It did happen in other places, and there are many countries which doesn't have, which have been declared as COVID-free, something like New Zealand and other places, right? So it's pretty good news that it will be getting contained soon. The last I want to talk about is the vaccines, which of course, the best thing to, anything which we would like to contain with any viral infection are the drugs. Drugs are always much helpful, but in this particular case, we don't have good drugs, and therefore vaccine is the only hope what we have. But the good thing is that many efforts are being made. There are over 50 vaccine trials which are going on currently. The most talked about obviously is the Oxford vaccine and something called Moderna vaccine. And we have seen the data on the Moderna vaccine, very good response, which you can see on your right side. That good response after the giving 25 microgram as well, a single injection, you get very good neutralization titer, almost as good as convalescent. We have also seen Lancet. Everybody was waiting for the Oxford data, just published, a day for yesterday in the Lancet, and again, very encouraging results, good response, both antibody as well as T cell response. There was also a paper in the same issue of Lancet from the Chinese vaccine. They are also trying. There are different vaccines they are trying in China, right? And our own biotech company, Bharat Biotech, has a Covaxin, which is kill vaccine, which is also trial is going on. So various trials are going on and good encouraging results. So it's very positive feeling at present that something is going to work in this area, but we need to wait and see until we see the challenge studies, which I don't know how they will plan, but obviously the phase 3 data one needs to see. But the results so far are very encouraging. So I would like to end my talk here. Thank you very much for listening to me. Do wear face mask. And 5 points to remember, avoid gathering, be brave and don't panic, clean, disinfect, and most importantly, educate yourself. Thank you. Thank you.